Good afternoon, students. Today we'll be discussing about the M.Com Pharmaceutical Chemistry crystallization. Unit 2, that is the unit operation crystallization. Okay. So this is somewhat different from the general term because it will be discussing a larger amount of topic. So see here the discussion. Crystallization method. So factors affecting crystallization we will discuss in this topic. So what is crystallization? You know very well. Crystallization is a process where atoms or molecules in a liquid dissolve in solid condition. They arrange themselves into a highly ordered solid structure called a crystal lattice. So this offers to minimize the energy state of the system. Suppose any solution or any compound is in a liquid condition or in a solid condition, solid is dissolved in some particular material. So they are going to arrange in particular order. Like they may be in a hexagonal order, they may be in a cuboidal structure, because they want to go for the energy minimization state. If they are in the just scattered in that substance or in the phase, then there will be liquidation, or there will be some energy entrapment. As a result, the product will not be stable. That's why to minimize the energy, the product want to be liquefied. See here the different diagram which is showing different crystals. There are different fashion of crystallization. Now there are four major steps of crystallization. Like based on the common description of the crystallization process, there are four steps. So first dissolution, then filtration, then supersaturation, then nucleation. So in case of substance, solid substance which will be dissolved in the particular solute, and the dissolved solute, the solute is dissolved in a suitable solvent by heating it until a saturated or nearly saturated solution is formed. And this step ensures that the solute molecules are dispersed and free to move. Suppose any products containing some impure substance and other additives, so they have to solubilize. Whether the impure substance will be separated out and the required substance will be solubilized. Okay? Then filtration, optional but common. So this process may be required for some of the substance, may not be. So if there are some insoluble impurities present in the solution, it is often filtered while hot to remove them. Or maybe sometime it had to be down to room temperature like this. This ensures that only the desired solution will crystallize. Otherwise, if there is impurity, they will not be crystalline. Then supersaturation. The solution is then cooled down or the solvent is evaporated carefully. This causes the solubility of the solution to decrease, leading to state of the supersaturation where the concentration of the solute exceeds its equilibrium solubility at a given temperature. Suppose the solution is in very hot condition, like sugar and water solution. They are in a hot condition, so they will not be crystallized. They have to be cooled down. And whenever they will be cooled down, you can see the sugar molecule will be separated out from the water. So the supersaturation is required. Sometimes the substance cannot be supersaturated, and we have to add some another substance or a particular substance which we require the crystallization. They have to insert it into the solid, and due to some partition attraction, they will be saturated. Then nucleation and crystal growth. So nucleation is when the solution becomes sufficiently supersaturated, small stable cluster of soluble molecules, that is nucleus, will begin to form. That is, first nucleus will form. So that is the small stable cluster of molecule or the crystal structure. That will be first will be separated out, but that will be begin to form as a crystal. So this can happen spontaneously, primarily nucleation, and can be initiated by the presence of seed crystal or impurities. That's the second nucleation. That means you have to give another seed. Suppose we wanting a copper sulfate solution, then we want to give the we have to give the copper sulfate from outside as a crystal. So as a result, the seed crystal will be there, and another substance which are solubilized in that matter, they will be crystalline in that particular structure. Now crystal growth. Small molecules from the solution then deposit onto the surface of this nucleus in a regular and repeating pattern, causing the crystal to remain size. So they may be in a hexagonal or cuboidal or in a any type of structure, they will be separated there. Okay. So next, going to the next step. Next, now after the crystal growth, so what is supersaturation? In case of supersaturation, they can be achieved by adding more substance to a solution than can normally be dissolved. This is a thermodynamically unstable state achieved most often in protein crystallography by vapor diffusion or other slow evaporation technique. So that means some of the unstable compound they have to be solubilized. And after the evaporation, the diffusion. Because in case of solution, when it is in a very dilute condition, they will not be solubilized. So there are different zones. See the zones of solubilization. There are metastable zone and nucleation zone and precipitation zone. So see the diagram. Here the green color represents the zone 1, blue color zone 2, and three, red color zone 3. The solution may not nucleate for a long time, but this zone will sustain growth. It is frequently necessary to add seed crystal. So in this phase, the metastable space, the nucleate will not form for a long time. They have to add it a seed crystal from the outside. Then there will be some nucleation. The protein crystal will nucleate or the molecule will crystallate and they will grow. Then precipitation zone, the protein do not nucleate but precipitate out solution. Then the molecule will be precipitated out by the substance. So having to the nucleation, so a phenomenon which is whereby a nucleus such as dust particle or a tiny seed crystal are commonly in protein crystallography, a small protein aggregate and start a crystallization process. So in case of nucleation, you have had some dust particle or some seed crystal, they will be aggregating the substance for formation of crystal. Nucleation process is very energy barrier, which is easier to overcome at higher level of supersaturation. So if the compound is not supersaturated, then nucleation will not be done. So that's why you have make the compound, you get it. Now common difficulties, there are some difficulties in case of nucleation, like if supersaturation is too high, too many nuclear form ends and overabundance of tiny crystals. Suppose the solution is too high, then multiple nuclei will form. So as a result, different tiny crystal will be overlapped among each other. Then you can separate. Suppose you are making some nanoparticle, or making some sphere. So if the all spheres are agglomerated in one space, then you cannot separate. Then you cannot separate the crystals. So the supersaturation in such a concentration from where a good crystal can be achieved. In supersaturated solution that don't experience spontaneous nucleation, crystal group often only occurs in the presence of adding another substance. Okay, just 1 second. So crystal growth often only occurs in the presence of added nuclei or seeds. So generally it doesn't occur without the seed crystal. So you have to add the outside. Going to the crystal growth stage. So adding single molecules to the surface of the nucleating lattice. Suppose this is a diagram, this is a hazy diagram that it is a lysosome molecule, the title of the tetragonal crystal. So see in the right side you can see the tetragonal structure. So the different crystal may be of different structure. So first of all you can add the seed crystal. They may be of tetragonal or hexagonal structure. And after the hexagonal structure has been added to the substance, another crystals will be coming. Okay. Next, we can see the macromolecule that is done in the University of Toledo. They have shown the image work, and whenever they will be added in a fashion, they will be showing some diagram. Now, major factors that affect crystallization, which is the most crucial for your study. Here, purity of proteins or purity of the substance, protein concentration, then starting condition, then precipitating agent, then temperature, then pH, then additives. Then additives like detergent, reducing agent, substrate, and cofactors, there are uses. Now, just 1 second. Now we will discuss major factors of the crystallization. That is the purity of the sample, then sample concentration, then is the starting condition, makeup of the sample solution, then precipitating agents or precipitant is there or not, then precipitating temperature and pH and additive should be there. Now, purity of the sample. How the purity of the sample is used here? Because if the sample is not so much pure, then the sample cannot be crystallized. Then sample concentration. If the sample is not concentrated at a certain amount, then you cannot get the sample to be crystallized. Then starting condition. Suppose you are just taking the sample in a cold condition. Then then the solubilization has not been done properly. As a result, crystallization will not be done. So there should be optimum starting condition like temperature or the viscosity, the solubility, everything should be maintained. Precipitating agent, precipitant. If some precipitating agent is added there, then there will be problem to supersaturation or the crystallization. That's why you have to remove out the precipitant. Then temperature. The temperature is also very important point. In case of some substance which will be crystalline out in salt condition or in the hot condition, some substance will be solubilized in condition. So that's why temperature is very much important. You have to keep some of the substance for overnight. You have to keep some of the substance in the freezing temperature, sometime in room temperature. That's why temperature is very important. Then pH. Suppose any substance which is in a basic condition, they will not be precipitated. Some substance which are in a acidic condition, they may not be precipitated. So that's why they should maintain a particular pH where the crystallization is be done. Next, coming to the additive, detergent, and reducing agent, substrate and cofactors. So if these substances are present in your current substance, then solubilization will not be done. Next, detergent, reducing agent, substrate, it should be removed. Okay. Then the precipitation and then the solution will be supersaturated in a good manner and they will be very good test. Now there are different diffusion methods, vapor diffusion, slow evaporation, dialysis, and now this is the interpreting the result of crystallization experiment where you can see clear drop, skin precipitate, then mushy crystals, macrocrystals, needle crystals, then plates crystal, road cluster, different structure of the crystals. So single crystal you can see there are different structure you can see quadral, hexagonal, trigonal. Okay. So these are the crystalline shape and the cuboidal. So these are the structure of different crystals. So that's the end of the crystallization. And, and there are some equipment which are helping crystallization. In case of industrial scale, there are larger amount which did not for study syllabus. Thank you.